because he has been in so many different countries, speaking so many different languages, and affiliated with I don't know how many universities in the world. So he is a guy originally from Hungary. He was studying at University of Szeged. He was in Leuven, my other university where I studied. He followed me in my footstep, then came to Stockholm in my footstep, then back to Hungary, where he was the head of the department, where now Szabolcs is the head of the department, in the cognitive science department of the university in Budapest. He is now he was in Jena, then in Magdeburg and in Jena. So this guy, I don't know, probably there are many more universities which I... Yes, yes. So he is a mover. And he is also interested in different cultures and vision. That's the only, I would say, thing which is not changing in him, vision. So Gyula will talk about vision and the peculiarities in vision with special regard to some cultural phenomenon also.
So can you hear me? Is the mic working? So thanks, Balázs, for the invitation. It's a great place, and the organization is really something which is recommendable to everyone. Thank you for being able to hear. Do you hear me now? Is it working? Okay. What I would like to talk about is my work a little bit about repetition-related phenomena, generally speaking. But when Balázs invited me to this symposium, I try to think a little bit from a different angle, and that's why these occasions are good. How can you interpret a little bit differently your own topic? Now, just a few last-minute additions. When I was listening to the talks, I realized that many of the things I would like to speak about has already been said. For instance, different learning can produce different connectivity and neuronal functioning, but the same behavior by Jean-Pierre Changeux's lecture. This I will refer to later. Then Pear said that the neurons get active in parts of early visual cortex where the moving stimuli are actually not yet there, but they will appear later, even prior to the stimulation. That's also something I believe we have something to do with. And then in one of the last lectures, we heard that the brain gets bored by repeated stimuli, being fatigue, and Professor Holland also talked a little bit about fatigue as well. So there are quite a few things which are, Annabelle Chen, schizophrenia, she just mentioned it, quoted. I could have said a few other things from her talk as well. Schizophrenia, disconnection syndrome, comes from Carl Friston. I also will talk a little bit about that. So basically, I can stop my lecture here. Everything was given. But if we want to insist a little bit and put it into a bigger frame, how you could interpret my data, and I will go back to this slide, is this so-called Worf's principle. Worf's principle says that your language determines your perception, basically. So you can go to the Wikipedia. How objects look to us to depend on how we sort or name them. That's the Wikipedia description. But basically, it's something they say how your language, how your learned communication methods determine your perception. And there are a few very well-known phenomena, like the categorical perception of colors. So if you are saying green and blue, then you are perceiving it in a categorical manner, while if you happen to be a Botswanian who is not, this, I see the nodding faces, so I don't go on, or the other race effect, when any other group effect actually is an example of this. And I just flash it up, and I will turn back to that in the end. Okay, temporal context. So far, Balázs mentioned that I'm a vision guy, and I always did vision in my life. And most of the time, we are presenting stimuli which are in isolation. However, in the everyday life, that's not the case. We are seeing always stimuli one after the other. There is eye movements all the time. The scenes are changing, so it's a far more complex situation. There are two basic paradigms which are trying to quantitatively test these kind of repetitions of the stimuli. Either you put two stimuli one after the other, repeating in pairs, or you put a longer sequence of stimuli. Let's talk now about stationary stimuli only, because if it's a moving stimulus, that's an entirely different business. So when it gets complicated. So when you are putting two stimuli one after the other, you can call one of them as a target. If something comes up behind, that can be a mask, then we talk about backward masking. If you put something in front, then you can call it as a mask. You talk about forward masking. You can call it as a priming situation, or you can call it as an adapter, and then you are talking about after effects. Now, I work most of these phenomena, but I just will pick up today adaptation-like phenomena as one example to you. What does adaptation mean, and why is it interesting? Neuronally, this is behavior. Neuronally, we really have no clue how the two neuronal and phenomenological behavioral things are related to each other, but let's say that repeating a stimulus leads to some kind of change in the perception. How is it manifest in the neuronal level? From the beginning of cognitive neurosciences, people realized that when you present twice the same stimulus, you get less activation as compared to presenting two different stimuli to the animal. And wherever we look in the brain, it happens. So this has been described in IT, infratemporal cortex, actually, at the beginning. Charles Gross as well had some studies on that from the 60s. These are delayed match-to-sample tasks, so when the monkey was engaged in the task, but actually they realized later that the task is not important. It can come out in anesthetized animals as well. So something like there is a repetition suppression, repetition-related suppression, and people interpreted it in many different ways. One interpretation was that it's a simple fatigue mechanism. Neurons are getting tired of what they have seen. There's a little problem with, there's a big problem with that. It's specific. So if I see the same face twice, like in this case, then I get adaptation. But if the neuron is presented an entirely different stimulus, then the activation remains high as well. Therefore, it cannot be just a synaptic fatigue mechanism. Although this theory is a bit outdated by now, it in a different form comes back recently. There are many other ideas. Most of them are bottom-up or local. They say that something happens in the neurons which changes the representation. This is such a big phenomenon, very reliable, nicely describable, that during the years, in many different methods, just like in EEG, MEG, or fMRI, has been replicated. In the fMRI, it got a big importance because people think now that by fMRI adaptation, presenting two stimuli one after the other and checking the Bolt signal reduction there, you can get a little bit below the resolution of the crappy 3 by 3 by 3 millimeter resolution of an average fMRI machine. So that got a big deal, and the mechanisms are important, although we don't understand them. That's what I would like to talk about. Okay, there are many studies, and if you look up in the literature, it's a big confusion. Many different names are given to the same phenomenon: repetition suppression or attenuation, SSA, stimulus-specific adaptation, or just simply adaptation. Some even call it neuronal priming, meaning that this is the neuronal correlate of priming. It's not true. Or repetition priming. Single cells, ERPs, fMRI adaptation. Just last week, I typed in different combinations into Medline to see what's going on. 1,000 hits for different combinations, and I just did a 5-minute search for that. Okay, let's talk about two repeated stimuli adaptation. If there is a prolonged exposure to one stimulus that leads to the change of your perception, just like in the case of the well-known motion after effect, the stationary pattern looks like moving in the other direction, or in the tilt after effect, where being presented to an oblique line for a long time, afterwards the vertical line looks tilted in the other direction a little bit. These are well-known. These are low-level and well-known adaptation and after effect phenomena. It's a marker of the plasticity and the learning on a very low level, on a very short time scale of the brain. That's why we like it. Now, this has been described for low-level stimuli for a very long time ago, but interestingly, for high-level stimuli, just like for objects or faces, one had to wait until like 2001 until Mike Webster, Dave Leopold described this for faces. Reason behind is that we needed a computer technique to morph together stimuli. So what we are doing here is a gradual change of stimulus A to stimulus B, and this would be like a 50% stimulus, just like in case of the analogy of the vertical line, oblique left, oblique right line. That was not possible before. So we have a morphing sequence like that for faces, and then this would be the 50% Angelina Hepburn in this case. Now, that's your perception. If I test it, most of you guys will say 50-50, it's either this or that person. However, when you are presented for a very long time to one of the extremes, it's important to fix it just here until I talk a little bit, fix it around the nose somewhere. When I present it long enough, and it can be like 40 seconds, like we have seen in one talk today, or yesterday, sorry, or a couple of seconds already is enough, actually. Then afterwards, the same, physically same stimulus, what you have seen before, flash, will look a bit more shifted towards the opposite direction. So it's a contrastive after effect. You would say, if I test it, most of you would say that this is more similar to Audrey Hepburn than to Angelina Jolie, although it's exactly physically the same stimulus. Now, this is a phase after effect, and that means that adaptation, prolonged exposure, biases your perception or your decision, whichever is your preferred expression for that. We didn't have a neuronal correlate for this thing for a very long time. Therefore, in a study, we tested it in an EEG experiment, and we found that a previous exposure to a previous phase for a long time biases your perception. It's a female-male decision in this case, so you had to make a female-male decision for faces, biases your decision, and parallel to that, the ERP components, they have one component which is reduced dramatically, the N170, which comes from this occipitotemporal region. In addition to that, this is a category-specific thing. It has nothing to do with the task, because if you are presenting a hand before, for a hand stimulus, you get the same activation reduction. But if I'm presenting a face before a hand or a hand before the face, I don't get any change or very little change in the ERP, meaning this is a category-specific effect happening around 150 millisecond post-stimulus presentation. And then we went on with this study, and I could talk about it a lot because I really like this phenomenon. It's really a robust phenomenon. But instead, I chose today as well to pick up a more exciting field, which excites me far more now, namely when you are presenting stimuli one after the other in a row. So stimuli in longer sequences. So instead of presenting one stimulus for a long time, let's present shorter time, more stimuli. That's the idea. In this case, people say that the successful recognition of the target depends on the contextual probability of stimulus occurrence. Now, what is contextual probability? The example. If I tell you that you are seeing someone in a swimsuit, then your prediction will be that that guy is laying in the sun here on the beach instead of doing something crazy like that. Because we learned it, meaning that we experienced it before a lot, that the two things, let's say, temperature and swimming suit come together. Yeah? So we learned something, and that's what we call contextual probability, meaning that our prior experiences determine our perception and shape our perception. Different experiences, therefore, can lead experiences, environments, can we call it culture, learning, or whatever, we don't know, leads to different contextual probabilities, therefore, different predictions about the brain. So if I were giving this lecture now in Jena, in my hometown in North Germany, I would face 90% blondine girls with blue eyes. I would not be surprised. If I got the same audience here in Singapore, I definitely would be surprised. And the other way around. So different predictions are made about the word. Can we pick it up? Can we somehow check it in the brain? Now, there is a big theory about it, which is controversial but lovely. Carl Friston, but Rao and Ballard before actually pronounced this theory, saying that there is a predictive coding network in your brain. It's a cascade of mechanisms. So it's a feedback, feed-forward, intimate interaction of many levels. But two is actually enough for that, referring to short or long-term loops. and they determine your perception. How? There is no such a thing like sensory input coming in, decision coming afterwards. The two things happen simultaneously. I already have a prediction. I mean, in our brain, we don't really have a blank sheet, right? So we always, suppose you had a blank sheet, you wouldn't have this top-down prediction, but that's never the case. Comes a sensory input, and that's immediately compared to your predictions. If they don't meet, like in this case, they don't match, then the error of your prediction, the red, is going to be forwarded into the system. Meaning that not the sensory stimulus is coded, but the error, how is it different from the predicted stimulus? Now, why is it good? Because we save energy. How? Next time you are presented, your predictions are going to be updated. The more you are presented to one stimulus, the more likely that is coming up. Therefore, the predicted stimulus representation is going to be more similar to the incoming input in this case, gradually. Eventually, through many repetitions, this leads to the reduction of the signal, the error. So repetition suppression reflects the reduction of the prediction error in this system. Yeah? What happens, and then it goes on, and eventually the neuron ceases to fire. What happens when a new stimulus comes up? Of course, the very same neuron is going to be still active for that, because that, again, leads to a high predictive error. Yeah? Predictive error high, response is high. Now, let me take a little break here. You guys, at least the younger generation, will know the person I'm looking for now. Tell me, who is playing in every of these movies? All of them. So there is one person playing in all of them. If you know it, don't tell the name, shout out loudly the hair color. Of course, one movie is not enough for this task. Oops. So, somewhere here, people used to say it. Who was that? Red hair, Julia Moore, yeah? So, it's still not messed up. So there are many pictures of Julia Moore coming up now. She's playing in every movie. You can say that she's a very attractive person. What happened in your brain when there was something in between? Repeated stimuli lowered neuronal responses. Unexpected stimulus, again, bumped it up, yeah? So what did I do with you? I made a little show about predictions. Let's predict someone who is Julia Moore in this case, and then something came up which didn't match your expectations, right? So now you see how the brain codes energy efficiently stimulations and stimuli. Let's go a little bit into the Julia Moore neuron, which doesn't exist, of course, but suppose there is something like that. The activation of this Julia Moore neuron would go down gradually until something else is coming up, and then for Julia Moore again back, right? Predictive coding, that's just to remind you, predictive coding theory says that the response of a particular area, Julia Moore neuron, in this case, is determined by the weighted input of the error and the predicted stimulation. So R is the expectation of the so-called representational units, these guys, leading to prediction, and A is the error. A is the error, the unexpected occurrence. So at the end of the series of the Julia Moore presentations, your representational unit activation in that was high, while the error was low. Exactly the other way around when I presented that other phase in between. Yeah? The weighted input of the two will determine, and the proportion of the weight will determine the neuronal activation for this Julia Moore neuron. Okay? So to sum it up, continuous matching of sensory bottom-up and predictive top-down information happens in your brain. Repetition slowly makes the predicted and incoming stimulus representation similar. This leads to an attenuated prediction error in the so-called error units of these predictor estimator areas. And this leads to a reduced response for repeated stimuli. Why is it interesting? Among others, it's a lovely, nice theory, Among others, it means that repetition suppression requires top-down interactions. It's not anymore a bottom-up mechanism or a local mechanism, and therefore it's very interesting to me, at least. That's what I'm studying a lot, how repetition suppression happens. In this case, this is a proof. If we can prove this, then it has to be a top-down effect. If I can disprove it, then it most probably is a bottom-up effect. So that's why it's interesting to us. What kind of proofs do we have for that? It's a lovely theory from Friston and from Rao-Ballard, and surprisingly, there were not many proofs of that for a very long time. One proof, which is interpreted as a signal of predictive coding, is the mismatch negativity. Now, you heard about mismatch negativity when it means that you present the same regular frequent sound interrupted by a rare other sound. Now, it can be like pitch, it can be intensity, it can be actually the lack of the stimulus as well. So you need to imagine like beep, 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 interrupted by the same or different but irregular sounds. Now, nice to prepare for talks all the time, because you realize that wherever I look, I see Professor Change's name. In my, this is honest, in my, how do you call literature, so in the collection of my PDF files, I had this paper for a very long time. But I just refer to it always, the Wacong et al. Paper. And then when I prepared the talk, I read what these guys said about mismatch negativity, really, because the title is what I'm saying now, that the mismatch negativity is interpreted and predictive. And I realized that I was surprised that Professor Change was an author on that. This is, I think we met like 10, 15 years ago. Last time on a conference, same thing happened at that time. So you can expect that still. So mismatch negativity is, together with this rare stimulation, you get a difference in a ERP component, which is between 100 and 300 milliseconds, quite fuzzy, but it's usually there in the ERP. Now, what are we saying? What do these people say is that the N1 signal, the reduction of that, is the prediction error signal, which is reduced by repetitions. Therefore, the novel signal reveals it, discloses the mismatch negativity. That's how they interpret, how people interpret the MMN results. Now, I have a lot of problems with this interpretation. First of all, it's an indirect theory, because they came up with the idea through studying schizophrenic patients. They argue that in schizophrenia, there is an abnormal experience-dependent plasticity, which might come together with a top-down impairment. So the fronto-posterior, let's call, interactions are impaired a little bit. And if that's so, let's say, if that's so, MMN shouldn't be there in schizophrenics. And indeed, there is a huge literature on that, which one cannot argue with. 120, 150 papers appeared by now on the fact that in schizophrenic patients, the mismatch negativity is lower as compared to control subjects. This is a mismatch negativity for one feature or for another feature. That's control and schizophrenia. So indeed, there is a big evidence for that, big body of evidence for that. But mismatch negativity is an automatic process. We know that it comes out even if you are sleeping. You don't need attention for that. Why would the brain need a top-down process for an automatic event, right? I don't think it's economical in one hand. On the other hand, we know that many predictive processes are conscious, require attention. Therefore, I believe there is a question mark. And repetition suppression is not really studied in schizophrenia. I found one study which says that the title says impaired repetition-related suppression in schizophrenia. And then you look into the paper, and that's partly true. For the hippocampus and for another area which I forgot is true, but for the FFA, for instance, they explicitly describe that we didn't find any modulation of the repetition suppression differently in schizophrenic patients as compared to healthy subjects. Therefore, there is a normal repetition suppression, at least in certain places in the brain. And by the next part of the talk, I will introduce a paradigm. In that paradigm, just mirror it now. We found, again, perfectly normal repetition suppression as well in 3 areas of the brain of the schizophrenic patients. And as well, a modulation was perfect. I will refer to that back later. And last but not least, I believe that the mismatch negativity is not a good paradigm. When you are talking about predicted and non-predicted, you would like to have a situation when they come up with equal probability. In MMN, the frequent stimuli are the predicted ones, the rare stimuli are the unexpected novel ones. Therefore, it's a confound. Predictability and probability are confounded with each other, meaning that the explanation might be simply probability, which is interesting still, but probability can explain these results. Now, there came another paradigm recently in 2008. Christoph Sommerfeld published a paper which made some uproar among us who worked a little bit on repetition suppression. I explain a bit detailed the paradigm once, because then I will use a lot this, so it's important that we understand it. He used phases, but it doesn't matter. He used repeated trials, S1 and S2 coming up one after the other. He used alternating trials, and he used target trials when the second or the first phase, depending on 50%, was very small. The subject had nothing to do with repetition or alternation, just had to press the button if I've seen a smaller phase. Okay? It's important that the task is orthogonal to any manipulation. Another thing, note that this phase and that phase are also different in size. This one is that one is also different in size. In order to get rid of the low-level feature changes, he always changed the size. Just sometimes the size change was far bigger. That was the target trial. Okay? So the subject did this fMRI experiment, and then afterwards he got rid of these trials, never analyzed that. Okay? So it's just really to keep the attention of the patients, subjects there. What did he do? He used a clever manipulation of the blocks. It's an fMRI, so I guess many of you were in the scanner. What do we mean under block? It's a bit untraditional in his understanding. You press the button and the scanner starts, that's the run, and within that run, he used 5 to 6, or in this case 3, I believe, different blocks. A block started with a screen, information screen. Please start your task. Prepare the starting for the task. That was the information screen. And then afterwards, for 40 trials, the stimuli had different probabilities. So there were either 40 trials coming with 60% repetition, 20% alternation, or the other way around, 60% alternation, 20% repetition. Clear? It's not recognizable. I ran already this paradigm. He did for 20. I ran already for 80 subjects. None of them reported that I realized the probability difference. It's not conscious, so it's impossible to recognize. I mean, if I would be in the scanner, I would know, but otherwise the naive subjects can't. Now, what he did, he compared the 2 different trials. So we had a trial type versus block type, 2 by 2. Yeah? And let's see what happens. What he found is that the gray bars are the bold signal for the alternating trials, and the empty bars are for the repeated trials. He found a lower response for the repeated trials than the alternating trials. So far, it's repetition suppression. But what he found is that this reduction of the response was significantly bigger in the repeated trials as compared to the alternating trials. That means that the repetition probability, the blocks, modulated repetition suppression, was larger in repeated blocks if the subjects expected the repetition as compared when they did not. Okay? Please note that I'm talking about, from now on, not prediction, expectation, repetition, probability. Okay? Because the 3 things are slightly different. Why is it a cool thing? Why was it an uproar? Because this really means that the reduction of the prediction error is related to repetition suppression. Therefore, the bottom-up explanations are not satisfactory. Now, I was surprised by this result, and I decided I replicate it. And I said that, okay, if it's true, then the predictions should be independent from position. And I was in this position invariance business from a long time, from monkey business. I recorded from single cells from monkeys. I worked with that a lot. Therefore, it was one idea was, let's try what happens if the subjects are fixating and the first and the second stimuli are not coming centrally always, like we always do, but they are coming peripherally, and one comes here, the other comes there. If it's a top-down effect, if it's really something related to top-down, you should get the identical repetition suppression modulation as compared to the non-overlapping situation. I had a paper before showing that repetition suppression itself is independent from the positions. So it doesn't matter if you are presenting the two stimuli like that or like this. You get repetition suppression. That was a previous paper. Yeah, this is something automatically. Never mind. So it put up already the main result. The actual finding is that you get this modulation in both situations. So when the two stimuli were overlapping, didn't matter left side, right side, you get a modulation of the probability, and you get a similar modulation if the two stimuli were non-overlapping. Of course, the signal is less, but the interaction of the two fractors, so mathematically it's an interaction of trial and block, that was always highly significant in both cases. And in every area I studied, surprisingly, even in one of the lateral occipital areas, not only in FFA or OFA. OFA, FFA we've heard today, OFA is occipital face area, another face area. Okay, so we replicated this once. That suggests that that's kind of high-level effect, because indeed the neuronal receptive fields don't matter anymore, so it has to be somewhere higher in the brain than, let's say, LOV4-ish level. So how high? We know that for faces there exists an illusion, which is called the Thatcher illusion. Do you see a difference between the two faces? What is the difference? Yeah, you know this illusion. It's far more striking, the difference, if I turn the two faces upside down, upright again, yeah? So what does the Thatcher illusion tell you? It tells that faces are not processed by parts. Faces are processed kind of configurally, holistically, whichever you prefer. Somehow you take the whole face in, and that's how you make decisions about these faces. Now, and they say that this step of face perception is far higher level than the lower, part-based steps of face perception. Yeah. So if it's a higher level, then comes the question if you really can use this manipulation of the faces for testing the expectation effects or probability effects. If repetition probability is really from higher level, then inversion should eliminate it. That was a great idea. We were 100% sure that inversion is going to eliminate the repetition probability effects. So we repeated again the same paradigm, except that in different blocks, different runs, faces were upright, sometimes they were upside down, but everything was otherwise identical as before. And to my surprise, for upright and for inverted faces, we got the same modulation. Here you see upright faces, alternating blocks, repeated blocks. Inverted faces, alternating block, repeated block. The difference between these two, the black and the red, is presented here as an index, and you see that for repeated blocks, the index is far higher, and it didn't matter which orientation. So that means that we know that, theorists say that this effect, the Thatcher effect and such holistic processing step happen in the FFA. Therefore, I would say that's in different areas as well, the same thing. Therefore, I would say that this repetition probability effect is originating from the post-configural stages of phase processing, even higher level. So it's somehow coming from higher level neurons. Okay. In the meantime, this was not chronological order as you have seen on the papers. In the meantime, I was in a conference in San Diego, and I sat down with my old friend and former supervisor, Rufen Vogels from Leuven, and he asked me, what am I doing? And I said that I'm replicating the Sommerfeld study because it's striking for me. And he said that, interesting, I'm replicating that too. And he said that he was so upset on the data he didn't want to believe. He's a monkey guy. He's recording from single cells in inferotemporal cortex. that he went into the business and showed the monkeys this same situation without task. So there was no task in the monkey thing. So macaques, and he recorded not only spiking activity but LFPs as well, IT, repeating and alternating trials, repeating and alternating blocks. The only difference from the previous things was that he used objects and fractals, such stimuli, because Ruffin insists that faces are bad stimuli. They are so special in the brain that we will never learn what's going on with faces. It's a great model, I would say. So he used these fractals and objects. And then he published the paper in cerebral cortex, and what he found was that, to cut it short, the reduction of the response in the two different blocks, that's what you see in red and orange and blue. The reduction of the response is identical, is very similar for the IT cells, meaning that there is no repetition probability modulation in the IT for objects. Therefore, Occam's razor, the simpler explanation needs to be valid. Simpler bottom-up local mechanisms are able to explain repetition suppression. He was attacked. Of course, the believers of predictive coding didn't want to say that something is wrong there. The monkeys are not human. The monkeys didn't pay attention. Objects are not faces. There were many other arguments, among others. And then we said with Ruffin, okay, let's do something together. Give me your stimuli, which is not an easy task because these are trial unique stimuli. So the monkey never saw the stimulus twice. Okay, you know, if you work with monkeys, you know that means 30,000 stimuli for a study. For fMRI, my poor students always record for this kind of things 300-400 stimuli and standardize them because that's the minimum. For a monkey, you need one magnitude more. So I asked for a stimuli. I got rid of from the stimuli everything which was an animal, a face-like thing, a body. So it's really just objects. So keep only objects like, I don't know, what is it, a flower or some kind of complex object over there. And in the human brain, we don't really know, but there is one assumed homologue for the IT, the LO, lateral occipital cortex, which should be here somewhere. So again, repeated the same boring paradigm one more time, repeated trials, alternating trials, repeated blocks, alternating blocks, the only difference being that I used objects this time. And from the LO, forget that, from the LO I got very similar repetition suppression for the two different blocks. That means that there is no effect of repetition probability, again supporting Ruffin's finding. Reviewer comes, says that that's nice, but can you show me please for faces that your subjects are not special? They do show the probability for faces, so I had to show it for faces, that for faces it does exist within the same subjects. Yeah? That's just a control experiment. So this is the important one. For objects, neither for fMRI nor for single cell, you get this, you don't get the difference. Now, what can be the difference between faces and objects? One difference is that the intertrial variance of the faces is far less than the intertrial variance of the objects I use. What intertrial variance means? Something like I standardize the faces a lot. So if I merge, overlap hundreds of faces on top of each other, you still see a face. Yeah? Features are identical. If I overlap the same amount, don't ask me how many was that now, but a lot, objects on top of each other, you get a noise. Therefore, look for a category which can be standardized as good as a face, but it's not a face. Not easy. I came up with the idea of chairs, IKEA catalog, downloading from the internet, chairs facing always you, same size, same viewpoint, sort of four-leg chairs, because there are, of course, different chairs. And then if you merge together the same amount, you still get an impression of a chair, right? Not perfect, I know, but still something. It's better than the objects. Cut it short again, you get repetition suppression in both blocks, no repetition probability effect either. Conclusion, it's not that. It's not the intertrial variance of stimuli, but it has to be something else. So what else? Faces are special. You know, that's what we always hear. Faces are special. But what does it mean actually to us? This is weird. We have a lot of experience, expertise, frequency of exposure, saliency and importance. These are all there for faces. Punchline is there. Find another category which is like that, letters. My daughter was 5, she started to learn writing and reading, and she was interested in the letters. And I realized that it's not a big difference between a face and a letter, right, those first 5 years, if you ignore inherited mechanisms, which are not there. So what we did, we replicated again the same thing, this repetition probability experiment, for letters. Now I used Roman letters, and after making a few unsuccessful pilot experiments, false fonts. So I took the letters, splitted them, but then I stacked them back in order to keep something of the gestalt of the stimuli. They were definitely novel. Now where to look in the brain? LO is an obvious candidate because that's for objects, but I know that there is a visual word form area in the brain, which is on the left side somewhere in the fusiform gyrus. However, I made a mistake. During the study, the localizer, the functional localizer experiment was done with single letters, not with words. And that turned out that that's a big difference because letters activated other area, which is really distinct, letter form area, a bit more occipital. So it's on the inferior occipital gyrus somewhere behind the visual word form area. Never mind, we have an area and we have two conditions. So I repeated everything again, alternating repeated trials blocks for these two types of stimuli. LO and the letter form area, in both areas, I got a huge repetition suppression, but only in the repeated blocks. If you see for the alternating blocks, you don't get anything. These are the Roman characters. Okay? So for the Roman characters, you get a repetition suppression only in the repeated blocks. Therefore, it's a strong modulation, just like for faces. What about in the same areas, same subjects? For the false fonts, no modulation whatsoever. Repetition suppression is independent of the block. Yeah? So that, what does it mean? It means that the effect depends, in my strict opinion, on prior experiences, on previous exposure, or something related to learning. This paradigm, the previous paradigm, was, after a while, got really boring. And second, it has a big confound. Expectation-related modulations, prediction-related modulations, are confounded with probability. Therefore, I try to separate the two. So I try to go on into the expectation versus repetition suppression separation. There are studies showing that. And I came up with a 2x2 design. If I can make it that some stimuli are repeated and expected or unexpected, and some stimuli are alternating and expected and unexpected, I can separate 3 possible mechanisms. One mechanism is that there is no such a thing like expectation suppression. Everything is probability. Yeah? You get a reduction of the response in both cases, both the expected and unexpected cases. Another possibility is that indeed, as people say, expectation suppression adds to repetition suppression, and then mathematically you get an interaction. Yeah? But it's also possible that repetition suppression and expectation suppression are independent of each other. Expectation reduces the responses. These are generally lower than these, but that's independent from repetition suppression because it equally happens in both cases. So to disentangle these 3, I came up with a design where the gender of the faces predicted repetition or alternation. For half of the subjects, female faces were repeated more. For other half of the subjects, the male faces were repeated. That doesn't matter. So for half of the subjects, a female face signaled immediately repetition with a higher probability as male faces, and for and the other way around. So male faces were more unexpected, more alternating than repeated. What I got is that In the FFA and the OFA, there is no interaction between the two factors. This is alternating repeated, expected, unexpected. You have a main effect. Both cases main effect, but no interaction between the two factors. Meaning that the two factors, repetition and expectation suppression, I skipped that, are independent from each other. And even in time, just to show nevertheless briefly, this is the Bolt signal modeled by FIR curve. And you see an index of repetition suppression that precedes in significance in time the significance of the expectation, whatever the different Bolt signal stages mean. We don't know that, but there is some strong suggestion for time as well. So summary, repetition probability effects on repetition suppression are present in humans for non-phase stimuli as well. It was a bit too short. It presents in every study, every area wherever we look, and it usually recommends suggests some higher level modulations. The effect is not phase specific or not area specific in the brain, what we are finding. Something else is able to explain its existence. What I believe is learning or experience, and that can, in my opinion, determine the predictive modulations. And in the last experiment, I showed that you can separate the two from each other, so we have a better tool even than the previous paradigm. Now, outlook. How can, let's go back to the first slide. How can you relate to the Worth's principle? This is replication of the first slide. My question is what one could study in a cross-cultural experiment or something like that. Is it merely a probability issue? Is it really just because we are presented with a statistically different environment in the, for different stimuli in this case? Then one could test it in different cultural environments. And the question would be that how the different contextual probabilities, what you are presented here and in Europe, in the different cultures, determine these repetition-related phenomena. Now, you could do this for faces and bodies, some high-level stimuli, and as a control, you could use objects as well, because there you wouldn't expect any difference. But obviously, it's a very, very interesting and unstudied field, actually, how similar face and character perception and processing in your brain is. Because if you are testing characters and you show the brain activations for a guy who is doing face research, they will say that these are face stimuli evoked activations in the brain. It's very similar. They are very much neighboring each other. So it's an exciting field. One idea says that the brain, when you are learning characters, recycles the areas of the brain which were originally for faces or before this experience and learning for faces. So that's something I will study and would be nice to do with other cultures, other statistical environments. And finally, what strikes me here is the variability of the stimulation if we are talking about faces, which is unique, I believe, to Singapore, really. I never saw that before. So different multicultural environment can be either volatile, meaning they are changing a lot, or they can be stable as well. Now, if you are testing the other race effect in Jena, my university city, there, to find someone who has some little experience with Asian faces is a nightmare. Because it's a closed city, kind of closed environment. I mean, not many of you guys are going there. Therefore, the exposure to Asian faces there is very little. It's a stable environment. When you go to Paris, it's already a different business, right? You come here, it's again a different ballpark. Therefore, I believe this might be the variability can be an issue, which is, I'm not a face perception guy. I work on faces, but it's for me a model only. But this can be studied, how the variability of the environment changes your neuronal representation for faces, in this case, in some cross-cultural studies, I believe, in the future. Thank you for your attention.
There was a last slide, but we don't know what it is.
That's Balaton, the big lake in Hungary, and that's a village where my father was born, and this is a village where I just recently bought a Ferienwohnung, holiday dacha, holiday house. It's a beautiful region, nice wine.
So, thank you, Gyula. It's great. Questions, Per?
So I should shout. I wouldn't shout at Gyula because you were suspicious with the predictive coding scheme, and obviously this is wrong already from a neurophysiological point of view. Namely, it postulates that the axons going from the higher order areas to the lower order areas are inhibitory. But there's no such axons. All axons connecting cortical areas are excitatory. So the model is, shall we say, biologically wrong.
Except if you are talking about connections within the same area.
Yeah, sure.
So
But that's not what we are.
That's something very unclear in the predictive coding network. Within the same area, you can imagine lower and higher order neurons, and then your argument is not true.
It is.
I'm protecting
between areas, it's correct.
No, it doesn't say that. But the predictive coding doesn't say whether it's in between or within areas.
No, it doesn't. That's right. So this is at least unclear. But the usual interpretation is, and if you speak to Dan Ballard, which I have done, that is also his interpretation. All right. That's good. Now, my question really is, did you present these faces back to back? That is, was there a gap with something in between it, or wasn't there? The reason why the reason why I'm asking is that we don't see objects initially, but we do see the changes in the luminance and color composition between a previous stimuli and the subsequent stimulus. But this is very clear if you present them back to back, but this effect might disappear to some extent if you present them with a gap.
500 milliseconds was the gap, 500, 500, 500, which is nothing if you do fMRI, but still argues against a bit you. What argues against you is that we found exactly the same effect if I put the S1 here and the S2 there. Therefore, on one retinal location, there was only one stimulus. So that's what I can say. And the size changed a little bit. So argument is there, but we do a few things against it.
You disprove.
I have a few questions. At the beginning, you talked about the position invariance for the repetition suppression. So for the repetition suppression you observed, are the two stimuli presented within the same the receptor field of the same neuron? Because the neuron has a larger receptor field. So if you present at the same receptor field, there is a possibility for position invariance.
Whether they were different positions in the same receptive field?
Right,
or well, they were real. Subjects were fixating in the middle, and one stimuli came 8 degrees on the left or 8 degrees on the right.
Yeah.
And they were like 3 degrees in radius. So they were very, very far away from each other contralaterally.
Contralateral is fine.
Therefore, I would say that except for neurons like MST,
yes, about 100 degrees receptor
not
field.
Even FFA, they were in separate receptive.
Does monkey has, did you get it from human subjects or from monkeys?
This is human study, everything, except Ruffenfogel's
so you cannot map the receptive field of those neurons, right? Because these are just human subjects. So,
okay. I didn't check it in the V1.
But V1 has much smaller receptive field, so
Yeah.
it won't cover the entire stimulus.
Usually, one just if you came up with that, so where you can measure the retinotopy in fMRI,
Right.
V1, V2 early visual areas,
Right.
you get usually a huge probability effect.
Okay.
Doesn't matter what.
I see. Well, yeah, because of the receptive field size difference along the cortical hierarchy. I'm curious about expectation, expectation suppression paradigm. Can you repeat those paradigm? Because it seems to me the repetition suppression or adaptation is actually very low level. It depends on the contrast adaptation, something very low level. It doesn't go through the prediction, but I don't know how you manipulate the expectation.
Expectation was that that was conscious. So subject came in,
Yeah.
and I told them that if it's going to be a female face, then with 75%, altogether 75% probability, you will see repetitions. And the subject was playing a little bit. If it was a male face, 75% alternations. That was for one subject. For the other subject, randomly, the other way around. Males predicted repetition, and females predicted alternation. So I equalized it across subjects, yeah.
Oh, the prediction manipulated by the probability of occurrence. It's
Exactly.
not the prediction of the future
S1
outcome of the stimulus.
Exactly. S1 predicted
I see.
probabilistically repetition or alternation.
Oh.
Yeah.
If you manipulate the percentage, are there any correlation between the two? Like you change from 70% to
No,
20%,
it's,
then
No, only two
sequentially?
left. It's long enough.
That will take a long time to do the experiment.
No, but it's a good idea. What we will do now, do a neutral level.
Okay.
Because whether it's suppression or novelty enhancement,
Yeah.
you don't know.
Okay. For this kind of experiment, do you change the stimulus size from?
Always.
You change it
Always.
between
S1
each-
and S2.
S1 and S2. I see. What's a behavioral task for them to do?
Say again?
What's a behavioral task?
Female, male, very easy.
Just to judge female or
Female,
male. Okay, I see.
Yeah, that comes from an acoustic paradigm. There is a guy called Floris de Lange in Holland, and from his lab there is an Anna Todorovic girl who made an experiment where an acoustic signal was repeated or alternating, exactly like high tone, repeated low tone,
Right.
alternating, and the subject's task was high-low.
I see.
That's a visual.
Did you morph between the female and the male and try to
case,
see
not.
the? So the accuracy is quite high, right? in this kind
Accuracy
of
was
experiment?
quite high,
Yeah.
yes. Was ceiling, actually, yeah.
Okay, gotcha. Did you measure the reaction time of the subjects?
Haha. Yes, but not analyzed yet.
Okay. Yeah, I'm interested to see the result. Maybe you can find priming effects from this. Okay. All right. I think that's all my.
Just one question. So you pre-queued the subjects in this case, but how can you compare then these results with Ruffin's results?
This is not to compare. This
Not to
is really
compare. Okay,
show
good,
that,
good. So it's on its
this
own.
Is
Good.
really, this is an entirely different, I believe, better paradigm to disentangle expectation from repetition.
What
rate.
happens if you don't queue the subjects beforehand?
See, Balázs, it's a really complicated what kind of prediction or expectation we are talking about. There are these three paradigms. The mismatch negativity, it comes out even if you are dead. The probability modulations of Sommerfeld, which doesn't need and doesn't evoke any conscious thing, and this kind of paradigm. No one yet tested which parts of the network we are testing with these paradigms, so I can't answer.
Jingi.
So when you say Wolffian hypothesis, it seems to me actually you are referring, or actually from your results, is new Wolffian hypothesis. That is, I mean, it's a nuance difference, but it's different. For example, in the Wolffian hypothesis, that it says that different languages will provide different worldview, right? But the new Wolffian hypothesis says that actually it's the usage of different languages that result in different representation, because the different language will impose different constraints on people's mental representation. And then that's the reason why people might have different cognition or interpretation. So the differences is not on the language itself, but the usage of the language, and which is a very important steps.
I would fully agree with you. I even would go further and say that language has no importance in that. That's a side effect of this thing that you are correlating language with certain facts. It is the statistical probability which determines then, which is determined by language. I mean, obviously more people see Chinese faces in China where people happen to speak Chinese as compared to a, you know what I mean.
Could you show the Ruffenvogel's slide once more? So what I'm after is that, if I remember correctly, the initial transient is not affected at all by repetition.
Yeah.
But after approximately 150 milliseconds or so, there is a decrease, right?
Yeah.
Now, your fMRI study is not directly related to the spiking, but it's related to, shall we say, local field potentials in the region to some extent. So you show that, well, there is maybe a general decrease in the local field potential, and then
this is spiking.
This is spiking, I know.
Spiking, but Ruffen did LFPs as well.
He did LFPs as well.
LFPs, it was there.
Yeah.
Local field potentials and spiking.
Yeah.
In the paper, you find exactly the same result.
Okay.
Yes,
That's
of course.
But it shows that the initial transient is of the same size. That's what his study shows, but the effect appears after 150 milliseconds. So there is some factor coming up in the network after 150
Absolutely. But it's always for IT. So the IT nurse, and actually with Gabor, I was going to ask him about it because when he had the lecture, because the initial part of the IT response carries different information as compared to the last. But IT neurons, when you present a stimulus, even for 10 milliseconds, fire for 500. Yeah. And most of the information, if you extract the information, is carried in the tail of the response, not in this transient high thing. That's always kind of there.
The transient is the change in color composition, luminance from the retina.
Level,
It can
be
so the otherwise is related to the actual object. Yes.
Mm-hmm.
So let me reflect on that. Yesterday I didn't have time to go into detail, but some of you might know the phenomenon called persistent firing. So what persistent firing is, in case you stimulate a neuron for quite a number of times, like 10 to 100 times repeatedly, and you stop the stimulation, your neuron still carries on firing for about several hundred milliseconds. Now, this phenomenon is known from rodents, and we actually identified 2 cell types which can do the same thing in the human. And more than that, there is a particular cell type which does it in a really interesting way, namely, you switch off the stimulation and your cell starts firing in an accelerating manner, then it stops for let's say 700-800 milliseconds, and it switches back for several hundred milliseconds, and you follow these cycles. Now, the other cell type does it without this cycling, so it's just a continuous firing for about a couple of 100 milliseconds. And interestingly, both of these cell types we implicated in these feed-forward networks. So. And none of them are pyramidal cells. I'm talking about GABAergic cells here, but they are instrumental in timing spikes in pyramidal neurons. So I think this is very relevant to you.
Thank you.
So, further questions? If not, thank you, Gyula, again.
Thank you.